Welcome to today's WALS lecture. I'm Dr. Jane Simoni. I'm the Associate Director for Behavioral and Social Sciences Research at the NIH and the Director of OBSSR, or the Office of Behavioral and Social Science Research. It's my absolute pleasure today to introduce our speaker, Dr. Jenny Tung. Dr. Tung is the Director of the Department of Primate Behavior and Evolution at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. And she's a Professor of Evolutionary Anthropology and Biology at Duke University. Dr. Tung received her Ph.D. in 2010 from Duke University, investigating the causes and consequences of genetic variation in wild baboons, advised by biology professors Susan Alberts and Gregory Ray. After a postdoc at the University of Chicago with human geneticist Yoav Gilad, she joined Duke's Department of Evolutionary Anthropology and the Duke Population Research Institute in 2012. While at Duke, Dr. Tung investigated the genetic and genomic consequences of social environmental variation in baboons, rhesus macaques, and other social mammals, as well as the role of behavior in primate hybridization. Dr. Tung also co-directs the Amboseli baboon research project, which started in 1971 and is one of the longest-running primate field sites in the world, in Kenya, correct? One of her major contributions to the project has been adding sophisticated DNA analyses to the decades of Amboseli observations and seeing new patterns emerge over time. Dr. Tung's research with macaques has shown that the stress of life at the bottom of the social ladder can alter the immune system, just as it does in baboons. The results provide evidence that low social status leads to disparities in health, not just the other way around, as some researchers have argued. In 2019, she was one of 26 people to win the prestigious MacArthur Genius Award, which recognizes exceptional originality, insight, and potential. In 2022, she founded the Department of Primate Behavior and Evolution at Max Planck, which focuses on the intersection of behavior, social structure, and genes. Dr. Tung's lab is particularly interested in how the social environment influences gene regulation, population genetic structure, and health and survival across the life course. Now for a few housekeeping items. For those interested in receiving CME credits for this lecture, the code is 50123. Also, for those watching, you can submit questions for the speaker by clicking the Send Live Feedback button on the video page. Others of you in the audience can go to the mic after the lecture to offer or ask any questions that you would like. One quick breaking news just came out. I'm very pleased to be able to say that just very recently our speaker was elected to the National Academy of Science. So congratulations on that, Dr. Tung. Without further ado, welcome, Dr. Tung.
Do I need to do anything to turn this on, or can you guys hear me okay? Oh, it sounds like okay. We're in business. Okay, great. Other than slides. Let me see if I can wake up my computer. All right, good. Thank you very much for that introduction and for the kind invitation to participate in the Wednesday, in the Wednesday afternoon lecture series, which I will do so shortly right after. Good, okay. It's a particular honor to be able to share some of our research here, which is the place that makes much of it possible. As Dr. Simoni mentioned, my research focuses on non-human social mammals, primarily our close living relatives, the other primates. But much of the research we do with humans in mind, because we think that by studying processes in our close living relatives, we may be able to shed some light into processes of human behavior that influence how we do over the life course. So perhaps to introduce that, I'll start by explaining my talk title, which comes from an essay written by is there anything I can do to keep it from echoing? Is that bothering you guys? Or,
It's bothering me. So
okay.
I'll go up and see.
We're good? Okay, all right. So this comes from an essay written by the 18th century British satirist Alexander Pope, an essay on man, Perhaps the most famous line of that essay is the one shown here. Know then thyself, presume not God to scan, the proper study of mankind is man. So what Pope was writing about in this piece is the fact that humans fall somewhere between beasts on one hand and God on the other, but are endowed with the logical facilities to be able to study the secrets of the natural world. And he thought, and he was exhorting the reader here, to focus those particular abilities on studying what he thought was the most appropriate subject for our own species, and that was to understand the human condition. However, I should also acknowledge the direct place where my talk owes an acknowledgement, which is to Cliff Jolly, an anthropologist, a professor of anthropology at New York University in New York City in the Center for the Study of Human Origins, who turned around this idea to make the argument that, yes, much of what we can learn about humans comes from studying human populations per se, but actually there's additional complementary information that we can get through either homology, shared descent, or analogy with the other living primates. He was particularly interested in the insights we could get, not necessarily from the great apes, our closest living relatives, but from a slightly more distant set of species. These are Asian and African monkeys here of the group of the Paianen group. That includes the macaques, as shown on the bottom left here, the baboons up here, and also charismatic species like the Highland gelada monkey of Ethiopia on the top left, and the mandrill on the bottom right from Western Central Africa. The point Cliff was making is that, like us, these are large-bodied, terrestrial, obligately social animals whose success in life depends in large part on how they interact with conspecifics who live together in the same group. Macaques and baboons in particular are ecological generalists that can be found across large geographical space. In fact, they cover ranges in the world that are second only to humans among the primates. And so Cliff imagined an analog, a non-human primate analog, to Alexander Pope, who he called Alexander Papio. There was no image of Alexander Papio in the original paper, but this is courtesy of AI-generated art, which will give us an idea today, and wrote in the beginning of this paper that, you know, to understand humans, we should also be looking outside of humans to give ourselves some context into where our species came from and to provide sources of inferential power that are difficult to gain in humans alone. So in other words, we should be studying baboons too, not just humans, even if our goal is to understand the human condition. And I think that's particularly relevant when we're talking about aspects of social behavior, the social environment, and how those things influence life outcomes. In other words, at least in much of the research on these topics, the social determinants of health. And so up here is something that's probably very familiar to most of you. It's aggregated evidence that our social environments, socioeconomic status, social integration, social isolation, social support, and aspects of our environments early in life during infancy and development have outsized impacts on how we do. In all of these cases, the y-axis shows measures of mortality or mortality risk or lifespan, and on the x-axis, measures of SES, social integration or early life adversity. You'll notice that the effect sizes here are very, very large. These predictive relationships are some of the most robust, largest predictors of human morbidity and human mortality that we know of. For instance, over here are some data from a paper by Raj Chetty et al., published a few years ago on American populations. The x-axis shows income at age 40, and the y-axis shows lifespan or expected age at death contingent on reaching age 40. Okay? And so perhaps what stands out most obviously is that women in all cases live longer on average than men. But the other thing that I hope you'll notice here is that the difference between the top percentiles and the bottom percentiles of income at age 40 translate on average to a difference of almost 10 years in lifespan. This is a very, very large effect size. And in addition, this is not a stepwise pattern where you go from a major impact on lifespan at a level of income that we might think about as conferring material deprivation, right? This is actually a gradient across this entire x-axis. Similarly, these are old data. This is from Alameda County, California, following individuals who were scored as either less or more socially integrated into their groups. And again, you can see the difference in the proportion surviving after about a decade of follow-up is very, very large. And finally, this is from the Adverse Childhood Experiences study, showing that the number of severe sources of disadvantage that kids face early in life reaches for decades into the future to influence, or at least to predict, age at death, right? This is 6, 7, 8 decades later in life. These are so consistent across different settings, populations, study types, that the social epidemiologist Lincoln Phelan suggested that social factors, such as socioeconomic status on one hand and aspects of affiliative social relationships and positive social relationships on the other, should be treated as what they called fundamental causes of disease that influence multiple disease outcomes through multiple mechanisms, where those particular outcomes and the mechanisms that determine them may vary across different sorts of settings. Indeed, there's some evidence for this too. These are parallel data from the same, in most cases, same studies that show differences in, sorry, not from the same studies, but from the same x-axes that show differences in the prevalence or odds ratios of different types of diseases across the socioeconomic status, social integration, and early life experience gradients. Okay. I think that this is a powerful idea, right, that social relationships are a fundamental cause of life outcomes. However, it's also a very difficult idea to grapple with when you're trying to do biological research in a wet lab. It's very hard to think about how to come up with a cellular model for a social relationship. And it's particularly slippery to think about studying disease mechanisms when the predictor variable of interest influences conditions that are as different from one another as bronchitis and diabetes and heart disease. So I think this is perhaps one of the reasons why these kinds of very, very strongly predictive factors in life outcomes have not gotten perhaps the attention that they might merit if you saw 10-year differences in expected lifespan for any individual cause of, say, an infectious disease. It's also very hard to think about what potential causal pathways might be, right? It's possible that differences in social status or social integration have direct consequences for morbidity and mortality outcomes. That's one possibility. Another possibility, though, is that both are independently outcomes of some third variable, like early life adversity or potentially like genetic differences between individuals. A third possibility is that this is not really a question for biologists per se, but that it's really a question for people who work in public policy, right? If these effects are entirely explained by differences in smoking behavior, then perhaps the research should focus not on physiological and molecular mechanisms, but on the effects of changing cigarette taxes. And so the possibility, the difficulty in resolving these causal pathways, I think is demonstrated by the fact that we've known about the social determinants of health for a very, very long time now. If I speak to colleagues of mine who are sociologists, they'll say, well, we know about the social integration stuff that you guys are working on in animals now, and we've known that since Durkheim, right? So this is an old idea, but one that still hasn't had much resolution in terms of causal pathways. So I'm going to come back down now to the idea that other animals are a source of insight here. And I think there are three ways in which that is the case. Animal models offer opportunities to do experiments that we cannot do in humans, either ethically or practically. We can actually intervene directly on the social environment and ask what ensues to try and establish strong inferential power for causality. In addition, humans live a very long time, and so it's very hard to follow individuals from cradle to the grave. So even when we go to our close non-human primate relatives, this becomes something that is possible, right? And it can be paired with very granular, direct observational data on the experiences that animals have from day to day. In other words, we can watch the baboons that I study on a near daily basis across their whole lives, something that is nearly impossible to imagine doing in human populations. And of course, comparative studies can provide very, very important insight into when and where particular types of social advantage or social adversity might matter. We work in all of these spaces, but today I'll focus on the top two. First, talking about work that we've now been doing for about a dozen years on experimental models of social hierarchy and chronic social stress in rhesus macaques. And then talking a bit about the work that we do in wild baboons in Kenya, where we're able to really watch processes unfold across the life course. So these are rhesus macaques, which I work on at the Emory National Primate Research Center outside Atlanta. This work has been collaborative for quite some time with Mark Wilson and Vas Mikopoulos at Emory University and with Luis Barrero at the University of Chicago. We study these animals because they form completely naturally, very clearly delineated and regularly enforced social status hierarchies. So they form ladder-like social status hierarchies that can remain stable in natural conditions for years or even intergenerationally. We also study them because, again, unlike in humans, it's possible to perturb those social hierarchies in a way that is largely controlled. So we can watch animals who are in uniform demographic conditions, groups of the same size, who all have access to healthcare, veterinary healthcare in this case, but all have healthcare access, none of whom smoke, none of whom drink, all of whom have the same diet, right? And so what we can do is take adult female rhesus macaques who have no prior social history with one another and who are not close relatives and serially introduce them one by one into a new social group. We make groups of 5 individuals each in the particular paradigm that I work in, and it turns out the ones who are first in tend to become the highest ranking individuals in their group. Individuals who insert afterwards, and we control order of introduction, just kind of insert in the dominance rank hierarchy, in the social status hierarchy, directly below the individuals who are already there. So we can form these groups, and then we can replicate them. And so typically when we've done these experiments, we're working with 9 or 10 replicate social groups, and then we can watch what ensues over the course of typically about a year or so. About 10 years ago, we were using this kind of paradigm, but we really wanted to get a very clear handle on causality. So we decided to add another manipulation whereby after a year, we took the individuals who were at the top of the hierarchy and put them together in a group. And then we put individuals who were second ranking in the previous hierarchies together in a group, and so on down the list. Because these are naturally very hierarchical animals, they just reform a new hierarchy, and then we can watch what happens next for the next year or so until we run out of money. Sorry, that's the truth. And so what this allows us to do is watch animals when they're high-ranking and then low-ranking, and when they're low-ranking and high-ranking, or stay high-ranking over the whole period, or stay low-ranking over the whole period, and that's what you're seeing here. The y-axis here is a measure of social status. It's an ELO rating, so it's actually the same kind of measure that is used to rank tennis players or chess players and so on. And what you're seeing are animals connected with themselves from what we call phase 1 versus phase 2 of the study. This is completely uncorrelated, and that is by design because we really want to know whether it's the animal's social status and the social environment that confers that's driving the outcomes we're interested in. Okay? So this is just what actually happens in practice. So what I'm showing, let's see. Here, there we go. Those are interactions. Those are competitive interactions between animals, and we watch them and we collect them, and as we collect them, we see who's winning and losing those interactions, and it gives us this linear hierarchy between individuals over time. And once the animals kind of figure it out, and then we are able to figure it out secondarily, those tend to stay stable over the entire course of our study. Okay, so we've used this kind of experimental manipulation to look at a variety of different types of outcomes. And I won't go through all of this here except to say that we think that our data provides pretty good evidence for a causal role of social status on several different dimensions that might be of interest to you, including aspects of prosocial behavior, affiliative social behavior, measures of personality, including anxiety and boldness. At the physiological level, HPA axis function and glucocorticoid physiology. And at the molecular level, differences in gene regulation within specific immune cell types circulating in the blood. And I'll just mention here that the reason we think this is causal is that we can see a relationship between the social status of the animals in our study and these outcome variables, both within phase, and then if we look at the difference in social status between phases, the delta, we also see that followed by a delta in the outcome variable. So I won't go into this in much detail here, because what I'd like to focus on specifically for the purpose of this talk is some of our work at the molecular level, at the genomic level, focusing on immune function. So this work was really catalyzed by the postdoctoral work of one of my former students, Noah Snyder-Mackler, who's now at Arizona State, and Joaquin Sanz, who worked with my collaborator, Luis Barrero, and is now at the University of Zaragoza in Spain. What we were doing was actually borrowing from a method that's used in human populations. We were going to our animals after these social status manipulations, and we were taking blood from them. And we were taking blood in multiple tubes, one that went directly into cell culture media, just sort of the minimum ingredients that cells need to live outside the body for a little while. And then we would collect another tube in parallel, where the tube also contained cell culture media, but was spiked with an additive. In this case, I'll show you data on lipopolysaccharides. So what we were doing here is mimicking infection by gram-negative bacteria ex vivo. Okay? Then we could culture these tubes in parallel. We ran them for 4 hours, and then sort of stop the reaction, collect the cells, and then use functional genomic approaches to measure the activity of all the expressed genes in those cells, both in sort of the control condition and the immune-stimulated condition. And I'm just sort of squishing down all of those results here on the right in a principal components analysis. So in this case, we had 45 females in this particular data set, and each of them is represented by 2 dots. One of them is their cells at baseline, and the other is their cells when they've been exposed to lipopolysaccharide, basically when they think they're being infected by bacteria. And it wasn't a surprise to us that this first principal component, this first axis of variation, simply correlates with whether or not those cells have seen this bacterial compound or not. It's exactly what would happen to our own cells if they were exposed to LPS. But what was very interesting to us and has catalyzed a lot of work since is this second axis of variation, PC2, is very strongly correlated with the social status of the donor individual at the time of sampling. So we can go from a sample eye view to this kind of scatter plot, which is a gene eye view. In each case here, each dot is a different gene that we were able to measure in the experiment. The x-axis shows the effect size of dominance rank on gene expression in the control condition. And the y-axis shows the effect size for dominance rank and gene expression in the LPS stimulated condition. This is generally positively correlated. So what it's saying is that if a gene is more active in high-ranking females at baseline, it also tends to be more active in high-ranking females when their cells are exposed to LPS. Okay? But we do see some exceptions to that, particularly in this point cloud down here. So these are all genes that don't show much of an effect of social status in cells that sort of model resting state, animals just sort of walking around in good health. But when they're faced with a pathogen cue, their responses differ. We do start seeing this new, previously masked effect of dominance rank. And what that means in this particular study was that low-ranking females tended to respond much more strongly to bacterial stimulation than high-ranking females. Genes in this cloud tended to be particularly involved in pro-inflammatory processes. Whereas if we looked at genes on the other side up here, these are the few genes that responded more strongly in high-ranking individuals. They tended to be involved in type 1 interferon signaling. So very, very grossly a difference between a sort of pro-inflammatory antibacterial response down here, and sort of the innate, for some of the first-line innate immune responses to virus on the top side. So my collaborator, Luis, who has an immunogenomic background, said, well, that's really interesting. And I said, that's great. It's really interesting. Why is it really interesting? And so he said, it's really interesting because we treated them with LPS. And LPS has a very, very well worked out signaling pathway within cells. LPS is recognized by toll-like receptor 4, typically expressed on monocytes and macrophages. And once it binds LPS, once TLR4 binds LPS, it can signal sort of through two alternative pathways, shown here on the left-hand side and the right-hand side, respectively. Genes that are shown in these pathways in blue are more highly expressed in low-status individuals, and genes in purple are more highly expressed in high-status individuals. So if you squint a little bit, you can see a little bit of what seems like polarization, even in intracellular signaling pathways after exposure to LPS. And what's interesting about this is that these pathways sort of converge on the bottom on these guys here. These are transcription factors, and these transcription factors act to produce a further regulatory cascade within cells. So what we were able to do is augment the gene expression data with data on what's called chromatin accessibility, or open chromatin profiles, which ask about the physical conformation of DNA within the cell. Most DNA is packaged quite tightly around histones, but the parts of the DNA that are open to transcription factor binding and therefore open to gene regulation tend to be a bit unwound. And what we found is that if we measured unwound DNA near genes that were more highly expressed in low-status individuals, they tended to be enriched for binding sites for NF-kappa B and related transcription factors. But if we looked at open chromatin near genes that are more highly expressed in high-ranking individuals on the right-hand side, we saw exactly the opposite pattern, enrichment for binding sites for interferon regulatory factors. So, in other words, our data suggests that the social environment is causal to changes in gene expression for hundreds to thousands of genes expressed in circulating immune cells. Not only does this matter at baseline, but it matters to the way that those cells are able to respond to other stressors, like mimics of pathogen infection. We think these changes are linked in part to that sort of physical structuring of DNA and its accessibility to transcription factor binding, that in turn get down to a granular enough level that they actually can alter the use of different signaling pathways within cells. So this is kind of where we were maybe, 6 or 7 years ago, and we did a little bit more work to delineate more of this, more kind of related stuff, responses to virus, responses to glucocorticoids, and so on. But there's a degree to which stopping at the level of gene regulation is still somewhat unsatisfactory. I can make the argument that what's going on in our immune cells probably matters to, to whole organism morbidity, but we can't really show it in these data. There's just a whole bunch of layers that get from here to the social determinants of health that we started with that aren't there yet. And we're not there yet now, but we were inspired a few years ago by work that some of you might know, the Pittsburgh cold studies, where Sheldon Cohen and colleagues took healthy volunteers and exposed them to live virus, rhinovirus, coronavirus, flu virus, and asked about the predictors of which individuals actually converted to clinical symptoms versus not. Okay, and this is a nice review. They did this sort of work for about 20 years or so. But in general, the pattern that they showed was that greater levels of psychological stress, including the stress of caregiving to chronically ill children, and also early life social disadvantage, was consistently a good predictor of who, when exposed to exactly the same amount of rhinovirus or coronavirus, actually got sick. Right? So in other words, it's not just exposure, but it's also susceptibility conditional on exposure. We thought this was a really, really interesting idea and something that might, in a version that we could implement, get us a little bit closer to the motivating questions we were interested in. We also had the advantage that in this case, we can't do this in humans, we could actually do the manipulation of the social factor itself as opposed to taking people as they walked into the study. In addition, with the rhesus macaques, we could use challenges that they had never seen before. So we could also look at both the naive response to pathogens and the secondary response to pathogens. So that's what we did. So this is another set of monkeys a number of years later where instead of giving them live flu virus, we actually just gave them flu vaccinations. And these are human pediatric quadrivalent influenza vaccinations, the 2019-2020 Sanofi vaccines. And we gave it to them both in phase 1 of the study and phase 2 of the study about a year later. We measured their gene expression and antibody titers before vaccination and then at serial time points after vaccination up until collecting antibody data at day 28 when we could measure variation and how well different individuals mounted a response to the vaccine. Okay. And you can see here that they respond to the vaccine. They make antibodies very, very well. So this is what we believe is basically never exposed to human flu or human flu antigens. This is day 28. These are vaccine-specific antibody titers. They actually maintain those antibody titers pretty well over the course of the year, maybe drop off just a little bit, and then that second shot is basically a booster effect. Okay? This shows a similar kind of pattern. This is the HAA titers, though, going from basically no ability to restrict flu to pretty good ability to restrict flu 28 days after they saw the vaccine. Okay, so if our gene expression data have anything to do with predicting antibody response, then we reasoned that gene expression has to predict the antibody response itself. And so that's what we asked first. These are just examples. These are sort of some cherry-picked examples of gene expression levels on day 1, so the day after the animals saw the vaccine, on the y-axis, and the fold change antibody response from day 28 to day 0. So more antibodies, right? And these are pretty strong relationships. These genes are chosen because they are involved in the innate immune response. The innate immune response to virus, weird, and other aspects of the antiviral response. But they are actually pretty representative of what we see overall. Our analysis indicates that just short of about 2,000 genes show a predictive relationship on day 1, the first day after inoculation and that 28-day response. And many of those genes are enriched in categories that we might expect, like the response to cytokines, like the innate immune response to virus, and so on. Okay? So then the question is, do these genes have anything to do with the genes that we annotated as responsive to the social environment? And that's what I'm showing you here. So again, here we're moving from a sort of sample level perspective to a gene level perspective. Every dot is a different gene here. And the x-axis shows the relationship between dominance rank and gene expression, social status and gene expression, the day after the vaccine was administered. And the y-axis shows the relationship between gene expression on day 1 and that antibody response on day 28 compared to day 0. And what I hope you'll see is that this is noisy, but it's actually pretty well correlated. It's pretty positively correlated, meaning that we have a lot of cases up here, perhaps more cases down here, but we have cases up here where high status drives higher expression of the gene, and higher expression of the gene predicts a more robust antibody response. And cases down here where low rank drives higher expression of the gene, but in those cases, higher expression leads to a smaller antibody response. So these, adaptive immunity predictive genes are also genes that tend to be very involved in the response to social cues in the same animals. Okay? And so, of course, what is potentially more interesting is to ask, well, then does social status itself affect the antibody response directly, right, perhaps through these intermediate regulatory pathways? And we find some evidence that it does. So if we look at phase 1 of the study, it's noisy, but we see a relationship between social status and the antibody response. It's weak, but we think it's probably there. However, when we look at phase 2, we see no association between social status at the time of sampling and the secondary antibody response, that booster-like effect. What's interesting about this is that the secondary response is almost completely predicted by where animals landed here. So how much they responded to that first introduction of something that looked like a flu virus, where they landed on day 28 here, basically explained how much they responded here. And so, again, quite noisy, but what's rather interesting to us is that if we look at their social status in phase 1, which is completely uncorrelated with phase 2, that has weak predictive power for the antibody response in phase 2, even though, as you recall, social status in phase 1 and social status in phase 2 are completely uncorrelated with one another. Okay? So what this suggests to us is the possibility that it's that first exposure that matters, and then that has a sort of a social history effect that follows these animals down the road. So that's sort of a snapshot of where we are on our rhesus macaque stuff, which we think is quite powerful for doing these kinds of repeated biological sampling, following social manipulation kind of stuff. But it's bad for stuff where you want to see what happens over the course of years. We've never been able to run these sorts of experiments more than a few years, and even that's pretty hard. So I think work in natural populations, like the baboons I'm about to tell you about, are actually much better for that, because these are cases where we can watch known individuals and measure very, very detailed characteristics about their experience, from cradle to grave. Okay, so these are the Amboseli baboons. They live in an ecosystem in the southern part of Kenya, near the border with Tanzania. And they are special baboons because they and their predecessors, their ancestors, have been under continuous study by the Amboseli Baboon Research Project since 1971. So the project as a whole is now in its 53rd year of continuous data collection. And what we mean by that is that animals in this population are recognized on site and are followed on a near daily basis, where information about their behavior, their environment, who they're interacting with, what they have to eat, and so on are routinely measured throughout their lifespans. I'm lucky enough to work on this project through the long-term foresight of Gene Altman and Stuart Altman, who founded this project in 1971. And one of the great joys of my professional life is that I continue to be able to co-direct this project with Beth Archie at the University of Notre Dame and Susan Alberts at Duke University. I would be remiss if I didn't acknowledge that almost all of the data I'm about to show you, certainly the field data, is to the credit of the expertise and professionalism of our long-term Kenya field team. I've highlighted 4 of them in particular here: Kinyo Waratere, Raphael Mutatua, Longita Sayodi, and Sarah Sayalel, who have been the senior observers at our site for many years. In fact, Raf just retired a few weeks ago after studying baboons in this ecosystem since 1981. So we owe them quite a lot. And this is a picture of Kinua showing you what we actually do day to day. These baboons are habituated to our presence. They neither consider us threats, nor do they consider us sources of food. And so our goal is for them to treat us like they would treat a tree, so we can just kind of be flies on their wall. There's a lot of metaphors, I know. Flies on the wall and write down what they're doing without actually affecting it. And so that's what he's doing right here. And this is the image of our current pedigree. Well, this is actually about 5 years out of date now, ranging from first generation down into the 9th generation. This is about just shy of 2,000 individuals that we've been able to follow, including their descendant generations. So what do we do with those data? Well, one thing we do is ask, well, what is true in humans, true in baboons? Can we derive insight into the patterns that have been reported in human populations by studying these animals? There have been 2 areas that we've paid a lot of attention to recently. One is related to what I showed you earlier, and social integration and social support and also social bond strength. I'll just mention here briefly, because I won't go into this in much detail, that there's also a strong relationship between social integration and social isolation in these animals, basically how much they're grooming with one another, and in other analyses, whether they have strong, stable friendships with a small number of individuals, and whether or not they're likely to survive in any given year. So this bar over here is a survival plot that separates the top quartile of most socially integrated females in our population from the bottom quartile, or the top quartile of most socially isolated individuals in our population. This translates to about 2 to 3 years difference in adult life expectancy in these animals. This is the biggest effect size that we'd identified for explaining differences in lifespan and survival until we looked at this thing, early life adversity. So, the ACEs approach is a very human-centered approach, right? It's actually not an approach that's used very often, even in other animals, even though people have been interested in early life effects for a long time. But we were spending a lot of time around social scientists, and we thought, well, we'll just give it a shot. I don't know, add them up and see what happens, right? And that's what we did. So we added up a simple sum of 6 sources of early life adversity, major sources of early life adversity, as we thought about them in baboons. And this is a survival plot, again, showing adult survival. So we're not talking about insults that lead directly to mortality, but rather insults sustained in the first few years of life that translate into measurable outcomes years to decades later. And this is going from what we think about as our silver spoon babies on the top, nothing in particularly bad happened to them, and then individuals who had 1 major source of early life adversity, 2, 3, and so on. This is the sort of plot where you think, I'm never going to get this clear of a result ever again in my career, and probably we shouldn't have had to do statistics to see this. But we just never thought about looking at it like that before. So the difference between these dark blue lines and these red lines is somewhere up to about a decade difference in adult life expectancy. And just to give you some context, many of us are used to sort of the dog-human translation, like 1 dog year is 7. Yes, thank you. 1 dog year is 7 human years. This is about a 1 to 3, 1 to 2 or 3 ratio. So if we were thinking about this in human terms, this would be a difference in life expectancy of about 20 to 30 years. It's huge. These are the major sources of early adversity we consider: early life maternal loss, early social status, maternal social connectedness versus social isolation, whether they have a competing younger sibling, right? Another sibling born within the shortest quartile of inter-birth intervals in our population, real sort of local density, their group size, which we think is an index of resource competition, and then this is a purely ecological factor, whether their first year was spent in severe drought. So many of these things have a social component, and in fact, many of them have, I think, explicitly a maternal component. I want to point out, this is the distribution, by the way, of the ACEs in our population. And I should say, because this is often not obvious, unlike in human populations where different sources of early life adversity tend to be fairly correlated with each other, the ones in our population are not very correlated with each other. In other words, they move fairly independently of one another, allowing us to somewhat dissect which of those sources matter to which particular outcomes. This matters not only to the individual experiencing early life adversity, it also has downstream effects on the composition of our population and on subsequent generations. Here is just a plot showing that age at death in these female baboons is the major predictor of how many kids they successfully leave behind. And those kids are affected by intergenerational effects. So in work led by Susan's former graduate student, Matthew Zippel, he said, okay, well, we know, right, that the kids of a mom who died early, who experienced maternal loss, that individual is likely to have a truncated lifespan. Do we know whether a kid who experienced maternal loss and then successfully grew up and had her own children then sees effects of her early life experience on her own kids? That makes sense? Yes, okay, thank you. And this is what he found. He said, controlling for F2's own early life experience, all those factors that I just showed you, if her mother, or its mother, this is both males and females, if its mother had lost her own mother early in life, which could have happened quite some time ago, the likelihood that this F2 individual survives to sort of the minimum age of reproductive maturation in this population is significantly reduced. Okay, so that's this red line here. This red line is, grandma died when my mother was young. We think that this can primarily be explained by long-lasting effects on maternal condition and probably don't require any sort of transgenerational epigenetic inheritance or anything like that. And I'm happy to tell you why if you're interested down the road. Okay, so these were big effect sizes. We were very interested in what's happening. We don't know what's happening. And so since those earlier observations, we've spent a very large proportion of our sort of our research energy on trying to tease apart the pathways through which this might work. And that's taken kind of an army of students and postdocs and collaborators around the world. These are some of the things that we've been able to look at, and primarily in females so far. So I focused on these demographic outcomes, adult and offspring mortality. Those are very clear effects or very clear correlates of early life adversity. We see differences in the social behavior of individuals who experienced a lot of early life adversity. They tend to be somewhat more socially isolated as adults, and depending on the nature of the early life disadvantage, sometimes achieve lower social status when they are adults. We see some effects, especially of resource deprivation on long bone growth. So I think that they're undergoing sort of a baboon analog of stunting. And we see physiological consequences, especially in glucocorticoid physiology. And I'm happy to talk about any of that, but for the purposes of the remainder of the talk, I'll just focus a little bit on what we've done looking again at genomic and molecular outcomes. This is inspired by the biological embedding framework, which might be familiar to many of you. This argues that the reason that early in life environment has this sort of long reach into adulthood and older age is because the environment is able to influence what's happening under the skin. It's affecting biological, physiological, and particularly developmental processes in those sensitive periods early in life. So systematic differences in the environment and experience then generate systematically different biological states upon exposure, which remain stable over time. And if so, those environments early in life have the capacity to influence trait variation across the life course. One of the specific biological pathways that has often been invoked to explain this kind of idea are changes in the epigenome, where the bulk of the research so far, especially in population context, has focused on one particular type of epigenetic mark, and that's the covalent addition of a methyl group to cytosine bases in the genome. So this is a change that can, in some contexts, change the way that genes are regulated. So the idea is that systematic differences in the environment change epigenetic marks in the genome. And for this to explain early life effects, they must therefore remain stable over time. And so the early environment has to be correlated with epigenetic marks measured in adulthood. And that's been shown in a variety of studies, including in humans, although there's always this consideration that we might be dealing with confounders, because early life environments often predict adult environments. And so this might be a direct effect of what individuals are experiencing at the time of sampling, rather than sort of a true long-term early life effect. In addition, if this is true, and I think this requirement has been paid a lot less attention to, then these changes must do something. There are 28 million CPG sites approximately in a human or a baboon genome, and many of those sites are functionally silent in terms of whether they're methylated or not. And so showing that differences in DNA methylation actually do something to gene regulation is, I think, an important requirement that we should start paying a lot of attention to in studies of social epigenetics or environmental epigenetics. Okay? And in fact, here's some work from now over a decade ago that shows if you go in and sort of surgically alter DNA methylation at particular sites from the promoter of HBB, only some of them lead to changes in gene expression and others do nothing at all. And so if we're going to prioritize things that we think are functionally important, then we have to be able to separate those two processes. So We mostly do observational work, as I mentioned, but occasionally we do have reason in Amboseli to want to collect blood samples, and so that's what I'm showing you here. This is actually Kinua again, looking as innocent as possible, early in the morning, on the savannah, while holding a meter-long blowgun in his right hand. He's just holding it, tucked away there. That contains an anesthetic-bearing dart, and so when the wind is still and nobody is looking, he'll just quickly raise the blowgun, breath propel it, and then we dart an animal of interest and are able to collect blood samples from them. We've done this in a very slow and torturous way over many decades, allowing us now to interrogate those same measures of early life experience and genome-wide DNA methylation in adulthood. This is work that was largely led by my former student, Jordan Anderson. And so in a study that just came out about a month ago, we looked at about 250 unique individuals in our population and about just under 500,000 CpG sites, targets of DNA methylation, spread across the genome in the context of cumulative early adversity, those individual sources of adversity I mentioned earlier, and also just overall sort of resource availability as indexed by habitat quality. These are pictures of the same part of our ecosystem taken about 15 years apart when the environment went through significant deterioration. So going straight to the results, what we find is that we do see persistent effects of early life, but only for individuals who are born in really low habitat quality environments. Okay, so this is a good way of looking at it. These are the effect sizes of early life adversity, of cumulative adversity for individuals born in low quality habitats versus high quality habitats. So if they experience maternal loss or low social status early in life or social isolation in early life, but they were in a resource-rich environment, we just don't see much of it. But if they experienced those things and were in an otherwise low-quality, deteriorated environment overall, we really see the effect, and that is detectable, often over a decade later in life. We can break down, as I mentioned earlier, these are largely uncorrelated predictor variables across individual predictor variables and count the number of individual sites in the genome associated with each of those predictors. And what we find overall is that it's really, in our case, measures of resource deprivation rather than sort of measures that we might think about as mapping more onto threat that leave this long-term early life signature. So drought, habitat quality, maternal loss, which probably has multiple dimensions to it in terms of the early life environment. So it's very exposure dependent. These different sources of early adversity are not interchangeable with one another. Nevertheless, I've also written that this signature is somewhat generalized. So this is a plot showing the number of sites associated with whatever's colored in black on the bottom. This is an upset plot. And many of those sites are shared. They're associated with both habitat quality and with early life drought. So in fact, this overlap is fourfold enriched over what we would expect by chance, and almost all of those patterns go in the same direction, right, where exposure to adversity points to the same difference in DNA methylation. So sites that are linked to one source of adversity are much more likely to be associated with a second source of adversity, but this is not just, we think, some sort of weird property of our data, because they do not overlap with age-associated sites. And they do not overlap with sort of technical batch effect associated sites. Finally, we wanted to ask about whether there's any evidence that these things matter to gene regulation, which is still really, really far away from, risk of disease late in life, but at least gets us the next step down the road. We think there's some evidence that there is. So what I'm showing you here are annotations from the epigenetics roadmap. It just sort of colors the genome based on whether that genome has high regulatory activity or low regulatory activity. And regions of the genome that are associated with social status also, habitat quality or drought, tend to be enriched in regions that are doing something, that we believe are doing something, whereas regions of the genome that are associated with age are not. Okay, so there's reason to believe that these things are active. And finally, in some work that I won't tell you anything about other than saying that we did it, we went on this long detour because we were so frustrated by not knowing whether these associations did anything, and designed a high-throughput genomics assay where we could manipulate only methylation status and then ask whether differences in methyl marks on otherwise identical sequences drove gene expression differently. Okay, and so that's what we did here. And we do find cases where early life drought-associated CPG sites, for instance, are active in yellow when they're unmethylated. They drive gene expression when they're unmethylated, but silence it when they're methylated. Okay, and so we actually have done this in, and again, work I won't talk about today, for basically the whole human genome now, and that data set is available for anybody who's trying to interpret their epigenetic data. What we find is that early life sites are not particularly more likely to drive gene expression than just sort of background things that you measure. But for those that are in active regulatory regions, the vast majority of them do show evidence for methylation dependence. It does matter whether or not they're methylated or not. So in sum, I hope that I've convinced you that there are ways in which studying species that are not our own species nevertheless helps tell us about our own species and helps resolve these perennial puzzles, these very, very difficult challenges in thinking about the social determinants of health, both through sort of experimental mechanisms that give us very detailed insight into causal directions and through life course studies that allow us to identify models for things that are important in humans that can be studied in complementary ways elsewhere. Going back to Alexander Papio, just to finish, this was part of the essay on man I showed you in the beginning, but a little bit further down, he says, go wondrous creature, mount where science guides, which I take as follow the science and go to use the resources and tools available to you to solve the problems that you need to solve. I don't know if that's what he actually meant, but that's the way we're going to interpret it here. And what I'll say here in this case is that, animal models have been incredibly important in the research mission of NIH in many different dimensions of what the portfolio does, right? There are animal models for cancer, and there are animal models for respiratory disease, and there are animal models for vaccines, and there has been much less of that work done in the context of social determinants. But I think there's an argument to be made that although the homologies and the analogies are never going to be perfect, you want every tool in the arsenal that you can bring to bear. And I hope that I may have moved you a little bit in that direction today. So with that, I'll just thank, there are many people who are involved in this work, some of whom I've mentioned. I also just want to particularly acknowledge that a number of the people who've led this work were trainees that were supported through the NRSA program and through the K99 program, and we could not have done all the work that we've done without that support. So thank you very much for that. And if there's time, I'll answer any questions.
Anyone who has questions can use the mics on either side. There is one. There is one question from online that I can start with. Let me say the CME code again. It's 50123. How does your research complement studies on chimps and gorillas in their relationship to human health? Similarly, do different kinds of primates offer different kinds of insights? If so, how?
Yeah. So chimpanzees and gorillas are much more closely related to us than the species that I talked about today. And so from the standpoint of direct comparisons to try to identify the basis for uniquely human traits, I think they're exactly the right sort of species to study. That being said, this is the argument that Cliff Jolly made in that paper I mentioned in the very beginning. There are reasons why going outside of the great apes can be very helpful. Some of them are purely just practical. Baboons and rhesus macaques can be kept very effectively in captivity, and that's why they have been the most important, some of the most important biomedical models in general among non-human primates. In field studies, because of their faster generation time, we also have much bigger sample sizes for them than is typical to collect for even very long-term studies, for example, of chimpanzees. From a sort of a biological standpoint, they also offer some similarities to humans that actually we don't share with other great apes. So chimpanzees, for instance, are much more ecologically specialized to deep to forested areas, whereas baboons are like us. and kind of like pigeons and, cockroaches and so they live everywhere, right? They do very well. They adapt to different circumstances. And so depending on the question you're asking, I think there's a high degree of appropriateness to looking at different systems. And of course, I know some people here think about marmosets, and there are people who study TT monkeys through the NIH portfolio. And those offer other opportunities that our guys don't, right? These guys don't pair bond. But other species do. So if you're interested in the sort of consequences of pair bonding and the consequences of the severing of pair bonds, for instance, baboons are the wrong place to look, but TT monkeys might be a great place to look. Yeah.
I'm curious if you've looked at any protective factors or better worded, anything that might reverse some of these effects that you see of early life stress or adversity.
Mm-hmm. So we've done some work on causal mediation analyses to try, really we've been doing, sometimes we're very much following in the footsteps, too, of people who've been working in humans. We're like, okay, we have early life adversity, and then they're dying later on, and what could be happening in between? And so that's been sort of the motivation to test a lot of those intermediates that I didn't have time to talk about today. But in the process of building those models, we've also tried to ask about buffering effects. And so for early life adversity, one of the things that, but both high social status later in life and greater social integration or stronger social bonds seem to have a moderating effect to that somewhat offset the effects of early life adversity. So it's not that we're trying to say, life is determined, in those first few years of life. There are aspects of the adult social environment that seem to matter a lot, both to outcomes that we measure concurrently and also to the magnitude of those early life effects.
How about experimentally? Any thoughts to run any sort of later life experiments?
We haven't tried that sort of thing. The life course stuff that we do in the baboons does not lend itself to experimentation. And the captive work we do, I think this is true for everybody who's tried to do captive work with social manipulations. It's really hard to do full life course studies. To my knowledge, and you guys would know perhaps better than me about this, the only experimental manipulation with full life course outcomes in rhesus macaques that's been done to date has been dietary restriction. And that's a huge effort. So I'd love to see it done, right? Because we have all these pieces that suggest it's going to have consequences that unfold across the life course, but we haven't been able to watch the same individuals in those circumstances for long enough to really say that with empirical data. And that's where you want to do your intervention work.
Okay, we're going to have to stop at this time. Is this mic on? I'd like us all to thank Dr. Tung one more time for an excellent talk presentation. And again, congratulations.